So today we start a conversation about microbial growth, and it's going to be a pretty cool story. We're going to talk about the control of microbial growth. We're going to talk about antimicrobial drugs, but we need to start first with basics of the dynamics of microbial growth and with the way we can quantify it and with the conditions that affect it. So first, since we're going to be talking about bacteria, let's make sure that we're all on the same page. I'm not going to torture you with sexual reproduction or mitosis. Basically, the whole story about reproduction is going to end right now. Bacteria divide via binary fission. That's asexual division. Essentially, what happens is that bacterial DNA is replicated. All the components of the cell are multiplied, doubled, and cell is separated into two halves. You can see in this image that nucleoid, the chromosome, is positioned in the way that origins of replication are facing the opposite poles of the cell. When chromosomes are properly oriented inside of dividing cell, a special protein called FTSZ forms a septum. Structurally, FTSZ is somewhat similar to a human protein called actin, protein that forms microfilaments in the human cell. What do you think is going to happen to bacterial cells if we will somehow knock out the gene for FTSZ protein? They won't be able to divide, exactly, because they won't be able to produce a septum. They won't be able to separate, to partition the cells. Does that make sense? If you would look at a table here at the bottom, you will see that each round of cell division doubles the number of cells. So after the first round of cell, if we start with one cell, after the first round, we're going to have 2. After the second, 4. After the third, 8. So you can fairly easily estimate, and not really estimate, but calculate how many bacterial cells you're going to have after, 10th round. It's going to be 2 in the power of 10. Does that make sense to you? So first round, 2 in the power of 1, second 2 in the power of 2, third 2 in the power of 3. You multiply by 2. Are we clear? Good. No, that's really important, because what often happens, I say, okay, first round, 2 cells, second, 4, third, and people say 6. No, it's not adding 2. It's multiplying by 2. So say if you start with 100 cells. After one round, how many you have? No. After one round, 200, because you multiply by 2. After second round, 400. After third, 800. So you multiply by 2. Okay? Now, every division takes time. Am I clear? That time that requires for the cell to divide is called a generation or doubling time. They vary greatly. For instance, generation time for E. coli, typical generation time for typical E. coli, in the typical 37-degree incubator on the typical TSA plate will be about 20 minutes. Which means you start with 1 cell, in 20 minutes 2, 40 minutes you have 4, in an hour you have 8. as far as I know, the fastest dividing bacteria in the lab that was identified was Campylobacter jejuni. It managed to divide every 30 minutes, 12 or 13, which is incredibly fast. However, if you are unlucky enough to study mycobacteria, like Mycobacterium leprae, causative agent of leprosy, its generation time in the lab is 14 days. To observe the colonies, you may have to wait for months. That's really a pain in the rear end to work with such microorganisms. Now, so far, I added to all my statements about generation time in the lab. Right? You can appreciate that generation time will depend on the growth conditions. We saw it when we isolated microorganisms from the environment. We used two different media, TSA and suborder agar. Suborder agar had lower pH than regular TSA medium, which inhibited bacterial growth. Does that make sense? Now, my question for you is very simple. If you take, for example, E. coli, typical bacteria that can live in a human gastrointestinal tract, and if you will compare the generation or doubling time of that bacteria in the lab and in the GI tract, in which conditions, in the lab or in the colon, this bacteria will grow faster? What do you think? You think in the colon. Generation time for E. coli in the colon is about 2.5 hours. In the lab it's 20 minutes. Now, can you explain it to me? Why it grows faster on the plate in the lab? Huh? Not only this, they pretty controlled, You don't
have cells fighting it, that it hinders growth?
There's no competition. Nutrients are abundant. It has nothing that restricts its growth. Does that make sense? And this is true for practically every microorganism. It will grow much faster in artificial conditions that we create in the lab. As soon as we can identify those, microorganisms will thrive in the artificial conditions where nutrients are abundant and competition is essentially zero. Does that make sense? Now, last thing before we wrap up with a couple of things before we wrap up with this story. So first, if I am about to tell you that we have two microorganisms, A and B, and A has shorter generation time than B. A has shorter generation time than B. Which one grows faster? It's simple. A, yeah. Just to make sure that we, on the same page. Second question. So you see the number here. It's pretty big. We're talking about 281 quadrillion. Okay? That's a lot. Like really a lot. This is how many cells can be produced from a single cell if its generation time is 30 minutes, and it will be growing for 24 hours. So the question, and that's about 1 pound of biomass. So why doesn't it happen? Why we do not get the massive amount of bacteria spilling from the plates when we inoculate them with environmental samples? Generation,
Not enough nutrients.
Exactly. They run out of food. And actually, it turns out that bacteria in their growth follow a certain dynamic pattern. Before we move on, do you understand what generation time is? Do you understand the idea of doubling every division? Last but not least, really important thing. On the exam, on this topic, not the exam that you're going to have on Thursday, but exam on this topic. It's probably going to be after the break, okay? There will be a question. You have so many cells in the beginning which have such and such generation time. How many cells are you going to have in 2 hours? Does that make sense? You understand what I'm asking? So say you have 1 cell generation time is 30 minutes. How many cells you have in 2 hours? So it's okay, 30 minutes, 2, an hour, 4, hour and a half, 8, 2 hours, 16. That's the answer. Am I clear? Good? Awesome. I may also ask you a different story. I'm going to ask you, we had so many cells in the beginning, and we had so many cells after, say, an hour. Does that make sense? You start with 1, in an hour you have 16. What's the generation time? So you have to count the number of generations and divide the overall time by the number of generations. That will give you generation time. Am I clear? I promise there will be nothing mathematically complicated. It's all going to be division or multiplication by 2. Numbers are going to be really simple, like 1, 10, 20, nothing too complex. Am I clear?
Yes.
Cool. So I mentioned the dynamic of the bacterial growth, and bacteria follow the growth curve. Not that they follow it, not that it's some kind of an order given to bacteria. It turns out that the growth dynamic follows that principle. So essentially, a bacterial growth curve can be represented by the number of cells on the y-axis, depending on the time of growth on the x-axis. Does that make sense? Which brings us a very interesting problem. So here on this graph, left bottom graph, you see the growth of a hypothetical microorganism which generation time is 1 hour. Am I clear? So you start with 1 cell. How many cells are you going to have in an hour? 2. That's your 2 cells. Now in 5 hours, you're going to have 32 cells, 2 to the power of 5. And in 10 hours, you're going to have 1,024 cells. If you want to plot the growth farther, in 20 hours, you're going to have 1,048,576 cells. If you ever build a graph, you can imagine how challenging it is to produce a graph which has 1 and 1 million on the same scale. Does that make sense? This graph will be practically useless. Instead, we use, when we plot the number of cells, we use so-called semi-logarithmic scale. Instead of plotting the number of cells on the y-axis, we plot the logarithm of a number of cells. Does that make sense? So I'm not gonna torture you with math, but basically if you want, we can talk about during the break. I can show you. It's pretty simple. I don't know where math-wise we all are, and I don't want to take the bread out of the Dr. Stitt's plate or anybody else from math department. So basically what happens, if you would look here, you will see that 10, 100, 1,000, and there will be 10,000, 100,000, 1 million. They will be all at the same distance. Does that make sense? So 10 in the power of 1 is 10. 10 in the power of 2 is 100. Am I clear? Not only this, another pretty cool thing is that logarithmic curve becomes a straight line. Does that make sense? When cells grow, their growth dynamic will be reflected by the straight line. Do you understand the advantages of semi-logarithmic scale? It allows us to easier plot the cell number. Now let's talk about the stages of the growth curve. The first stage is called lag phase. This is the lag phase. Lag means cells do not grow, or you can't detect them. Why it can happen? After inoculation of liquid medium, cells are stressed. Believe me, they are. Every time you perform a transfer of bacterial cells from one sample to another medium, every transfer, every change, even minimal change of the environment imposes its normal. Stress means it's a normal change in the environment. Cells will respond, usually, with temporarily arresting the division. Does that make sense? They basically need to figure out the conditions. Are we clear? Once they figured it out, they start to grow. This stage is called logarithmic, or another name for it is exponential growth. At this point, cells are actively dividing. They're practically not dying yet. Okay? So very few dying cells, a lot of nutrients, and very little waste. Eventually, as you can imagine, the number of old cells in biology for the process of aging, the common word that is used is senescence. Just a fancy word. Basically aging. So number of aged old cells increases, and those old cells start to die. Does that make sense? And at some point, the number of dying cells will be equal to the number of newly divided cells. So the population will exist in the stable numbers. Now remember, the curve that we're looking at, the curve at this image, is the logarithm of living cells. Does that make sense? So at this point, number of living cells at the given moment is constant. This is what happens, for instance, in the countries, in the developed countries. When the birth rate declines and equals the death rate, which happens, for instance, in Europe, where the birth rate in the population is basically equal to the death rate. So the population of European countries practically stays the same. Does that make sense? Good? Cool. Okay. So, at this point, the amount of nutrients slowly declines. The amount of waste accumulates. This is called stationary. If you keep the population in the medium long enough, it will enter the death phase. During the death phase, the number of cells that are dying are above the number of cells that are dividing. Nutrients become very sparse and wastes accumulate. Can population, can the cells, can this community of bacteria, can still be salvaged? Turns out they salvage themselves. Many bacterial species produce so-called persisters. In the death phase, when environmental conditions change very unfavorably, some cells manage to dramatically slow the cellular metabolism, entering, I would compare it to the stage of anabiosis. You know, like in the sci-fi movies, when astronauts fly to, like, I don't know, another star system, they're placed in the artificial sleep with a very slow metabolism. Okay? So persisters do practically the same. They're not forming spores, they cells. Okay? Sometimes it's not another name, different type of cells that can be formed under the unwelcoming environmental conditions are called viable but not non-culturable. Short VNC cells. Viable non-culturable cells. Does that make sense? So these cells still can survive. They basically living. They're just really, really slow living. And they present a huge problem in clinical practice, especially in the procedures that are associated with the implantation of the sterile prosthetics. When people receive, say, a hip transplant, prosthetic hip, it's really hard to keep the artificial structure, the artificial hip, absolutely 100% sterile. No cells whatsoever. This is why people are often placed on antibiotic therapy right after the surgery to make sure that there are no bacteria growing in the hip. Am I clear? However, that facilitates the formation of persister cells. That pressure, antibiotic pressure, that constantly kills bacterial cells, may actually favor the formation of persisters that are really hard to kill by antibiotics, almost impossible. And when the patient is taken off the antibiotics, persister cells do not experience that terrible environmental pressure anymore. They speed up their metabolism, and you have at your hands the full bacterial infection of the hip. It's really a big problem. Does that make sense? Now, a quick question. Cells in the environment, in your gut, on your skin, in the soil, in which phase of the growth curve they exist? Stationary, of course. Because if they would exist in a death phase, they would all die. If they would exist in logarithmic phase, especially the ones in your gut, you would explode in a matter of hours. So it can only be stationary. Does that make sense? Now, why do we care so much about such seemingly fundamental and detached and theoretical thing as the growth curve? We use bacteria in many industrial applications. And in many of those industrial applications, bacteria produce, useful stuff. Useful for us. For bacteria, that useful stuff is often waste. So if you think about alcohol, it's a bacterial poop. Okay? If you think about all the organic solvents that some fermenting bacteria produce, it's also a bacterial poop. It's not just bacterial poop. It's not just a waste. This waste can actually lead to bacteria dying. Make sense? So one of the challenges of fermentation technology is to maintain the industrial bacterial populations at the logarithmic phase. So industry prefers logarithmic phase. So how can you do that? How can you maintain bacterial cells in the logarithmic phase? The answer is chemostat. Chemostat, my engineering skills are limited to basically putting a nail in the 2x4. So chemostat has a pipe that feeds bacteria with necessary nutrients, and a special system that constantly removes effluent without affecting the population. So basically it drains the old medium with all the waste that we need, maintaining the favorable growth conditions, maintaining the abundance of nutrients and low levels of waste. Before we move on, any questions? What you need to know. You have to be able to describe the growth curve. There will be no essay questions, obviously, but if I ask you to characterize a specific stage, you should be able to recognize it. Does that make sense? What happens during stationary? What happens during death phase? You have to be able to recognize the description. You need to know what persisters are, and you need to know what is the stage necessary for industrial bacterial applications, which is logarithmic. Are we good? Now, expanding on the growth curve, how can we know how many cells we have? You understand the question? How do we count bacterial cells? They're really tiny, and it's really hard to say which ones are dead and which ones are not. Okay, so the most direct way is called a direct cell count that uses so-called Petrov Hauser chamber right here. It's basically a hemocytometer. I don't know if anyone ever used one. Some of you may have, yeah. So here's what you do. Do you see this? It's really cool technique. You see this metal surfaces. These metal surfaces are covered with a very fine and almost invisible grid of crevices. Okay. Now you put a cover slip right over here, and then you load your bacterial sample. It is usually pre-stained, something like crystal violet, methylene blue. You load it here and here. Your bacterial sample spreads under the cover slip. You put your Petrov Hauser chamber on the microscope, bring it to focus, and this is what you see. You start looking at the grid. Does that make sense? And then say you focus on this particular square. You see that inside of that square, there are 25 really, really tiny small squares, like this or this. So you select, let's say, usually people select 5 squares, 4 in the quarters and 1 in the center, and they focus on these squares and they count the number of bacteria. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, 10 bacteria. You have 10 cells in the square. You following me? So you count the number of bacteria in all 5, divided by 5, that's your average. And then you have a formula, which, like, manufacturers provided. We had it hanging on the wall in the lab. So we knew if you count these squares, you multiply the number by, say, I don't know, 5,000. And that's the number of bacterial or whatever cells in 1 microliter. That's it. Now, what are the advantages? It is super simple. There's no sophisticated equipment involved. It is relatively fast. I mean, time-consuming, yeah, you gotta have a dedicated person who will sit and count. Does that make sense? We did that with certain blood cells. You can do blood cells. So generally speaking, when you have two samples in one chamber, it takes about 5 minutes, I want to say, to count two samples. It's pretty quick. You have a counter, so basically like click, you know. But it's really tedious, okay? And there is a quite distinct operator bias. What I mean by that? So when you look down the microscope and you see something, it is your interpretation whether it is a cell or it is a piece of junk. And my interpretation can be different from my Kayla interpretation. Does that make sense? So if we're talking about counting 5 cells, that may really affect the results. If we're talking about counting 105 cells, if we got slightly different interpretations and she's got 103 and I got 105, that's not big of a difference. Makes sense? But if she's got 3 and I got 5, that's 60% different. That's a lot. You see the idea here? So, but it's convenient, and we call it quick and dirty. Okay. On top of that, you can use the dyes that will allow you to distinguish between, say, all cells and dead cells. Okay. So the dye that will be taken only by dead cells, or the dye that will be taken only by live cells. So direct cell count allows you to tell apart living cells and the dead cells, which is necessary for building a proper growth curve. Am I clear? Now, Coulter counter is the machine that makes the cell counting automatic. Here, in this image, you can see Coulter counter, that tube, submerged into a very diluted, and I want to highlight it, very diluted bacterial sample. Now, if you would look at the schematics of the Coulter counter head, you will see that in the head there is an aperture, okay, opening. Now, this opening has 2 electrodes that measure electrical resistance across the opening. Every time the cell is passing through the opening, electrical resistance changes. Does that make sense? Change in the electrical resistance can be measured. So basically what happens, think about it this way. It's a good analogy. So imagine that you are observing an entrance into the building, but you're not seeing the doors and you're not seeing people. You only see a shadow of someone who walks through the door. Does that make sense? You see, every time you see a shadow, you can say somebody walked in. Does that make sense to you? You understand what I'm saying? You don't really need to see an actual person. All you need to see is a shadow. So this device allows you to see a shadow of a cell, a change in the electrical resistance. Now, my first question for you is, can it distinguish live cells from dead cells? No. It doesn't really matter. That makes sense? Which cell moves through, who cares? Second question. Why we must use very diluted bacterial cultures when we use Coulter counter?
If there's too many bacterial cells in the solution, it'll just look like it's always measuring a different rate than it would have normally? Like, there's too many cells, they're going to always be passing through, and you'll never not have it?
Not really.
Okay.
Now think about if you have people walking through the, if you have, like, very infrequently people are walking through the door. It's probably going to be one person every time, right?
Mm-hmm.
But if you have a crowd running to the doors,
Okay.
you see, there is a good chance in concentrated solutions, there's a good chance that instead of one cell, several cells will pass through the aperture at the same time. Does that make sense?
Yes.
They will still give you the same signal. You see, when people walk through, you will still see a shadow, but this will be a shadow from several cells. Am I clear? So, advantage here, it's fast, automatic, you don't really need to do anything. The disadvantages, it cannot distinguish live cells from dead cells, and you gotta use diluted specimen. Are we clear? When we will be done with count, I'll tell you kind of what you can expect. Okay. Now, we talked about it, just the golden standard for counting live cells is called viable cell count. So say this is your culture sample. You prepare serial dilutions, just as we did with a pour plate, remember? Just as we did with a pour plate. You don't have to use pour plate, you can use spread plate. But the point is, you prepare serial dilutions, you put them on the plate, you count the colonies, and you get an exact number of bacterial cell, well, exact number of colony forming units per mil. This is by far the most, I don't want to say the most accurate though. There is a subject, you know, there are errors associated with the dilution, with shaking, sampling, and everything. But it's probably the best way to estimate the number of bacterial cells as close as possible. And I'm talking live. Does that make sense to you? You can imagine that this method can be very time consuming. So imagine that you are doing a growth curve. How many samples, how many tubes with the liquid culture should you grow? Minimum. For statistics. Minimum. Classic number.
35.
3. 3 is a charm. 35. 30, I would kill myself over 35. No. Because let's count. You have 3 tubes. You take samples. Let's say you take samples every hour. And let's say you take growth over 8-hour span. So we're talking, well, 7. 0 and 7 more. So 8 samples. Let's round it up. Let's make it 10 samples. So 10 time points, 3 tubes. Every time point, you have to take 3 samples from each tube, which gives you 90 samples like this. 90 vials with a bacterial specimen, and for each of these 90 vials, you have to prepare serial dilution, probably 7 or 8 dilutions, which is a lot. So it comes down to several hundred microtubes, which you will have to pour on several hundred plates. That's a lot. There are ways to kind of reduce the clutter a little bit, but we're still talking about, like, there are 6 well plates, so there's a plate, size about of human palm, with a small 6 wells, like, imagine tiny petri dish, okay? You can do this, but it's a lot. A lot of work, a lot of tedious work. but it gives you the best estimate of your growth curve. Does that make sense?
Yeah.
That's the best. Any questions? There's actually another method that we're going to use at some point. If you have sample that is really, really diluted, like super diluted, like water from Lake Erie, what you can do, you can run this water through a filter which will collect only bacteria, put it in a special medium, and see colonies growing on the filter. Or you can basically slap the filter against the agar plate and see, you know, how many colonies, how many cells were filtered from, say, 1 liter of water or 10 liters of lake water. Make sense to you? We're going to do it. Now, these two methods, one is super convenient, super fast, with a lot of disadvantages. It's called turbidimetry. Turbid means cloudy. Turbidimetry measures the cloudiness of the cell culture. Am I clear? The principle is super simple. This picture on top, I'm going to call it 1, shows you the clear liquid medium. Can you see that? Clear liquid medium. Now, this liquid medium will allow a certain amount of light to go through. When bacteria start to grow, they obstruct the light path, and much less light is coming through. So you compare the medium with the growth to the medium without. Are we clear? We basically measure the light absorption or optical density of the sample. This method is very fast, very quantitative. Now, can it tell apart live and dead cells? No, it cannot. But what is convenient, it's quick. It works really well during the log phase, because during the log phase you practically have no dead cells. Okay, all cells are alive. Does that make sense? And what you can do, you can run turbidimetry along with the viable cell count for your specific cell in your specific conditions, and you're going to have sort of a reference point. You will know that, I don't know, optical density of 0.2 is equal to, I don't know, 1 million colony forming units per milliliter. Optical density of 0.3 is 1 billion colony forming units per mil. Does that make sense? You can make a reference. Which brings us to the last method that we're going to talk about. It's called the most probable number method. Now let me introduce the application, why we're going to use it. Think about, again, water in Lake Erie, for instance. Are there a lot of bacteria there?
Yes.
Yes. But they are very diluted. There are a lot of bacteria, but the volume of the water is also enormous. Does that make sense? So, say you want to check if Mentor Beach Park is suitable for swimming. So you go on the beach and you sample water in different locations, blah, 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 bring it to the lab. Does that make sense? And then you take a little 200 microliters of that water and put it on the agar plate. You have pretty good chances not to have any bacterial cells in these 200 microliters because sample is diluted. You see what I'm trying to say? You may or may not catch it. So it's not really relevant. Basically, as far as I remember, according to CDC, you must not swim in the water if the number of coliform bacteria, coliforms like E. coli, fecal bacteria, is above 400 cells per 100 milliliters, your chances are basically zero to catch a single bacteria in 200 microliters. Does that make sense? So you would play a lottery. So instead, we use the most probable number method. Here's how it works. You prepare, bless you, 3 sets of 5 tubes with a growth medium. Can you see that? Now, you inoculate one set with 10 milliliters of your water sample, another set with 1 milliliter of your water sample, and the third set with 0.1 milliliter of your water sample. Which inoculum, which water sample is more likely to have bacteria? 10 mil, 1 mil, or 0.1? 10 mil. Do we understand? So a couple of days later, you look at the results. And what you see is that all 5 10 mil samples showed bacterial growth. Not surprising, right? It's like all 10 milliliter samples contain the bacteria. Are you with me? You look at 1 mil sample and you see that only 2 tubes were positive. Right? Which means in 2 out of 5 samples, there were some bacteria. And then you look at the last set, and there is no growth. Meaning that none of your 0.1 mil sample actually had bacterial cells in them, because they were too small. Am I clear? What you do next? You go to a special magic book that contains a magic table, and you look at the magic table. And magic table tells you that if you have 5 positive in 10 mil, 2 positive in 1 mil, and 0 positive in 0.1, your most probable number is 49 bacterial cells per 100 milliliter. Now, before we move on to analyzing a couple of other numbers, let's figure out why we call it a most probable number. It turns out that this method is heavily based on statistical analysis. And this result tells you, first of all, this result explicitly says we have no idea how many cells exactly you have in your sample. Am I clear? But most likely, you have 49 cells in 100 mil. Does that make sense to you? Can there actually be 52? Yes. Or 47? Yeah. Absolutely. But we don't know. The highest chance is that you have 49. You see what I'm trying to say? Basically, it's kind of, you take a random sample of people from Cleveland. Is there a chance, say you take 10 people, is there a chance that there's going to be one person who is a Saints fan? Well, yeah, there's a chance, but it's pretty small, right? Now, you take a sample of 1,000 people from Cleveland, chances increase, but still, you may have sample randomly 1,000 people from Cleveland, and none of them is going to be rooting for Saints. Or maybe all 1,000 will be rooting. But those are quite improbable outcomes. Does that make sense? So when we give a result of the most probable number analysis, we also give confidence limits. For our particular result, the confidence limits are 17 to 130. Now what it means, the very basic level. means that actually our results with a 95% probability lies in that range. So our result is from 17 to 130 with the highest probability of it to be 49. Does that make sense? You understand? So say I tell Brian, my result is 49 cells per 100 mils, and the confidence limits are 17 to 130. So how Brian can interpret it? He thinks, okay, so 49 is most likely. Can there be 100? Yeah. Much lower chances, but it can be 100. Can there be 20? Yeah, but much lower chances. You understand the idea? 49 is the most probable, but the result can be in the range from 17 to 130. We clear? Now, we're going to mention this, okay, and then we're going to go over the quiz, and then we're going to take a little break. So I just want to say a few words, because we're going to talk about like cycles, biogeochemical cycles, and I don't want to start that too early. Chemical composition of the cell. Mostly water. Second go proteins, nucleic acids, carbohydrates, lipids. You don't need to know the numbers. You don't even need to know the order. What I really, really want you to understand, which chemical elements you're going to find in the main biological molecules. What I mean, I ask you, what is the chemical composition of carbohydrates? Tell me carbon, hydrogen, and oxygen. What is the chemical composition of lipids? Carbon, hydrogen, and oxygen. If we throw phospholipids, then it's phosphorus as well. What's chemical composition of proteins? Nitrogen, sulfur, carbon, hydrogen, and oxygen. DNA. Nitrogen, carbon, hydrogen, and oxygen. So if I ask you a question, in which biological molecule you can find phosphorus, and I give you a list of phospholipids, DNA, RNA, and protein, you tell me phospholipids. In which biological molecule you can find sulfur? Protein, DNA, RNA, triglycerides, you tell me protein. Am I clear?
Yeah.
Awesome. Now, first let's get out the term that confuses students often, and I try to explain it as clear as possible, which is growth factor essential nutrient. So growth factor, let's talk about definition. Growth factor or essential nutrient. It's the chemical that cannot be synthesized by an organism and must be provided exogenously, must be present in the growth medium. Does that make sense to you? Say you have a bacterium that cannot synthesize an amino acid tryptophan. You have to provide tryptophan in the growth medium. Am I clear? Or you have a bacterium that cannot synthesize vitamin B1. So you have to produce this vitamin in the growth medium. Or bacterium cannot synthesize nucleotides, all of them. So all 4 nucleotides, or say A, G, T, and C, well, you have to provide A, G, T, C, and U bases, let's say bases, in the growth medium. Am I clear? So please understand that examples that I give here, they are examples. Do you understand? So growth factor essential nutrient can be anything. Does that make sense? Anything. The definition is it's the molecule that cannot be synthesized by a microorganism. We clear? Nutrients can be divided into organic and inorganic. Inorganic nutrients are usually ions like sodium, potassium, chloride, magnesium, calcium. Organic nutrients are amino acids, usually vitamins, stuff like that. And one thing that I really want to highlight before we wrap it up is that there is a lot of recycling of chemicals between microorganisms and the environment, meaning that this main chemical elements, nitrogen, sulfur, carbon, they cycle through. Carbon dioxide, is that organic or inorganic molecule? Inorganic, very good. It's inorganic molecule, but when it goes through Calvin-Benson cycle and photosynthesis, inorganic carbon is incorporated into organic compounds. It becomes organic. Does that make sense? When you burn wood, is that organic? Yeah, wood is organic. But when you burn that organic carbon, you release CO2, which is inorganic now. Does that make sense? So when you have proteins, or say you have urea in the urine, okay, which is nitrogen inside of the organic compound, urea gets metabolized by bacteria and ammonia is released. This is sometimes why stench, okay? Ammonia is inorganic nitrogen. So bacteria constantly recycle and move, and they convert inorganic compounds into organic and organic into inorganic. This happens during biogeochemical cycles, which we're going to talk about on Thursday after the exam. Deal? Okay. Let me pause this.